Ex vivo lung perfusion, EVLP, is a process of evaluating and preparing donor lungs outside of the body prior transplant surgery.
I never felt this good in my whole life. That's the awesome thing. So I'm able to. It gave me a second chance at life. So it's really great. And exercising and just enjoying being alive.
EVLP with the Ex Vivo Perfusion System (XPS) represents the next frontier in transplant medicine.
Kyle would not have survived till today. We were losing him at the time the lungs became available.
EVLP with XPS in Steen solution is a valuable and FDA-approved resource to assess organs and potentially improve some that would have been turned down because of quality.
And in his particular case, there were lungs that nobody else really was interested in because they weren't optimized. And so because we had the availability of ex vivo lung perfusion, we went and looked at those lungs, and my colleague went to do that. One of the surgeons went to look at those lungs and actually procured them, brought them back, and although they weren't perfect, we were able to optimize them on the ex vivo lung perfusion device.
EVLP, expanding the donor pool for lung transplantation. More than 70% of donor lungs worldwide are deemed unusable and not used for transplantation.
There are currently about 35 million Americans suffering from chronic lung disease, and every year we do less than 2,000 lung transplants.
I always say that we already proved that this technology is safe. We've proven that. So I also tell them, we're not going to put you at any extra risk. So why not? We're not going to put you at any extra risk, and at the same time, we're going to be able to choose from a larger pool. So there's no downside to this. There's no downside. So why not do it?
And the lung basically can heal itself in many ways on the rig. So we begin to actually expand the lung, put the lung on a breathing machine, we're perfusing it, we cool it slightly, and then we rewarm it, and then we test how much oxygen can actually be put into the fluid through the lung.
It's a technology in which we can look at lungs that at first look may not be transplantable.
EVLP is a method to assess lung viability of donated lungs. EVLP aims to expand the potential donor pool, improve function of the lung that might be deemed unsuitable, and ultimately decrease inflammation and rejection. The EVLP technique is used in Europe and Canada, and approximately 300 lungs have been transplanted using the method. The process involves a 3 to 6 hour period during which the donated lungs are placed inside a sterile plastic dome attached to a ventilator, pump, and filters. The lungs are maintained at normal body temperature and perfused with a bloodless solution that contains nutrients, proteins, and oxygen. This can reverse lung injury and remove excess water in the lung. During the process, the lung function is evaluated continuously on key indicators. For example, how the lungs exchange oxygen, airway pressure, and lung compliance. When they are determined to be suitable, the lungs are transplanted into a waiting patient.
I've had this transplant for almost 4 years now, and it's a huge difference from where I started. Where before I had trouble doing anything, and as the years progressed, things got more and more hard to do. And so for me to have this is it's just like a different person. I guess I haven't felt this good since I was probably in my 30s. And so anybody has the chance, they have to do it. They really should. Don't be skeptical like I was. Dr. Griffith was just, I don't know, he's just such a nice person. I don't know how else to say it. I'm just grateful for him that he made this work.
They're just happy to be alive, and I'm happy to have delivered an organ that we otherwise wouldn't have used. It's incredible. Think about that, We have the power of life just because of this wonderful new tool, you know, and we don't want to withhold it. I mean, we want every transplant center to have this opportunity, you know, to use it, because it should be standard of care very soon.
The advantages of EVLP are: lungs are oxygenated during the EVLP process, more time for preparation, more time to make sure the lungs meet acceptability criteria, increase the organ donor pool, increase the utilization of donor lungs.
And that's how ex vivo lung perfusion has you know, filled up a perfect spot for us, because by having this technology available, we are able to move faster in terms of donor evaluation for specific recipients.
You know, it's an additional opportunity, and while we can't promise anything, it's a possibility, and then it's all voluntary. So we sort of, I think we sort of covered it, and sometimes I do have to say that sometimes these patients, especially in your situation, they're very anything, please, you know, and the parents are whatever you can do, you know.
The Novel Lung Trial, sponsored by Ex Vivo Perfusion, is the first prospective and multicenter clinical trial designed to evaluate the safety of ex vivo lung perfusion, EVLP, as a method to screen and identify good quality grafts from lungs that have been unused or rejected for transplantation.
So this project, the Novel Trial and the XPS device with Ex Vivo, really represented an unbelievable opportunity for our state to be able to evaluate lungs that are marginal at the outset.
And this is a tremendous opportunity. It's a tremendous opportunity because this is the first time that a group of centers get together to follow a specific protocol in ex vivo lung perfusion. So it's not anymore about one center, it's not anymore about different ways of doing ex vivo, it's about many centers doing exactly the same way.
And that was the exciting part of the trial, because there was no difference between the standard organ and the EVLP organ. So, I mean, that's a big win. We weren't trying to be better. We took organs that every other center turned down, okay, and we made them work as well as the organs that everybody wanted.
So the trial itself, what we need to do in the United States is that any new technologies or drugs need to be approved by the Food and Drug Administration, or the FDA here.
The study started in 2011 and initially involved 6 centers in the U.S. In August 2014, the FDA approved the method of using XPS with Steen solution for EVLP of initially unacceptable donated lungs at body temperature. XPS with Steen solution is the only FDA-approved system for EVLP of marginal lungs. The trial is ongoing in the USA with the goal of 110 patients in each arm. The one-year follow-up of the study showed that EVLP with XPS and Steen solution is a safe diagnostic tool to increase the percentage of transplanted lungs by screening the unused donor pool. If you are interested in participating or to learn more about the Novel Extension trial, please contact your hospital or referring center.
But this is the first major step, and we can recover maybe 30% more lungs if we use this in a systematic way in this country for lung donation. And that would be an incredible advance, you know, if we could get 1/3 more people per year transplanted.
A life is worth a lot. And so I think that type of opportunity using this technology is fantastic, and allows us to grow the number of patients that can receive a lung transplant.
For us, at the end of the day, it's about saving lives. Unfortunately, you know, donation, something tragic happens. And to take that tragedy and turn it into a miracle for somebody else is really, you know, the magic of what happens here.